Welcome back once again in your biology class. This is Nowsin and you are watching Educational Support. If you like this video, then press a subscribe button and the bell icon right next to it. And you have turned on all the notifications on your phone so that you don't miss a single video. So please watching till the end of my video. In my previous video, I was sharing tuberculosis part 2. In that class, we will learn pathophysiology of tuberculosis, diagnosis of tuberculosis, and treatment of tuberculosis. So if anyone missed my tuberculosis part 2 video, then visit my YouTube channel Educational Support, and link of this video is given in my description box below. So go for it and subscribe my channel Educational Support. So today we are going to learn about most important topic of molecular basis of inheritance, that is DNA as a genetic material. In this class we have studied topic DNA is a genetic material. Various experiments like Griffith prove that DNA could be genetic material. But only one experiment of Hershey and Chase ultimately proved that DNA was the genetic material. So let us see the topic DNA is a genetic material. Ever wonder why your eyes are a certain color, or why you look like your mom or dad? Well, that's because of your genetic material. Genetic material known as deoxyribonucleic acid, that is DNA. DNA is the hereditary material found in the nucleus of eukaryotic cells, both animal and plant, and the cytoplasm of prokaryotic cells, that is bacteria, and that determines the composition of the organism. So next we are going to learn about what is genetic material. Do you know what is genetic material? The genetic material of a cell or an organism refers to those materials found in the nucleus, mitochondria, and cytoplasm, which play a fundamental role in determining the structure and nature of cell substances, and also capable of self-propagating and variation. So, protein, RNA and DNA were taught as genetic material, but many experiments suggest that DNA as genetic material rather than protein and RNA. So genes were known to be carried on chromosomes, and the chromosomes were found to consist of DNA and protein. Next, I am going to discuss characteristics and properties of genetic material. Few requirements for a genetic material. The following are the properties and functions which should be fulfilled by a substance if it has to qualify as genetic material. Number 1 is it should be present in every cell. Number 2 is it must contain all the biologically useful information in a stable form. Number 3 is it should be able to store information in the coded form for the control of biological functions of the cells, and it also to express its information. Number 4 is it should show diversity corresponding to the variety existing in the organisms. Number 5 property is it should be able to replicate precisely and then faithfully pass over its true copies to the successive generation. Number 6 is it should be capable of variations, that is recombination and mutation. Further, such variations must be stable and inheritable. Number 7 character is the genetic material should be able to generate its one kind and also new kind of molecules. Last but not the least, number 8 is it should be capable of differential expression so that the various parts of an organism may acquire specific form, structure and functions in spite of having the same genetic material. So, obviously, one would look for a macromolecule to satisfy the complex functions of genetic material present in the genes. Originally, the proteins were thought to play the role of genetic material because they show enormous variety due to various combinations of their 20 constituent amino acids. However, they have no mechanism for replication, that is duplication, which is the most important feature of genetic material. Hence, they don't qualify to act as the genetic material. However, it turned out on the basis of experimental evidence that DNA, that is deoxyribonucleic acid, is the genetic material. So, DNA shows a wide structural variety because the 4 types of nucleotide units it is composed of may be arranged in an endless manner in its molecules. It also fulfills all the requirements of the genetic material mentioned above. So viewers, if you enjoyed this video or learned anything, then press a like button and don't forget to subscribe. If you anyone missed my previous videos, then visit my YouTube channel Educational Support, and link of these videos are given in my description box below. So go for it and subscribe my channel. Next is identification of genetic material. One of the first studies that ultimately led to the identification of DNA as genetic material was done by Frederick Griffith, involving the bacterium Streptococcus pneumoniae in 1928 and concluded in 1952 with the study of Hershey and Chase. Next, I would like to discuss Frederick Griffith experiment. Frederick Griffith used two stains that are distinguishable by the appearance of their colonies when grown in laboratory cultures. In one stain, a normal virulent type, the cells are enclosed in a polysaccharide capsule, and capsule protects the bacterium from defense system of the body, giving colonies a smooth appearance, and this stain is labeled as S and able to cause death. In the other stain, a mutant non-virulent type, that is non-lethal, the polysaccharide coat is absent, giving colonies a rough appearance when grown in a petri dish and does not cause disease, and this stain is called R. So, Griffith's conclusions or steps in the experiments. Griffith injected mice with living R-type bacteria, and the mice were not affected, and after a while the bacteria disappeared from the animal's bloodstream. He also injected mice with living H-type bacteria. The mice died, and H-type bacteria could be isolated from their blood. So Griffith killed some virulent cells by boiling them and injected the heat-killed cells into mice. The mice survived, showing that the cells don't cause death. However, mice injected with a mixture of heat-killed virulent cells and live non-virulent cells did not live. Live cells could be recovered from the dead mice. The cells gave smooth colonies and were virulent on subsequent injection. Somehow, the cell debris of the boiled S cells had converted the live R cells into live S cells. The process is called transformation. So, do you know what is transformation? Transformation is a change in genotype caused when cells take up foreign material. Next, I would like to discuss the Hershey-Chase experiments. The Hershey-Chase experiment were a series of experiments conducted in 1952 by Alfred Hershey and Matthew Chase that help to confirm that DNA is the genetic material. Harshey and Chase and subsequent discoveries all served to prove that DNA is the hereditary material. Next, we are going to focus on bacteriophage. This is a picture of bacteriophage. Did you know what is bacteriophage? Bacteriophage is a virus that infects bacteria. When bacteriophage infect bacterial cells, they produce more viruses, and these viruses are released when the bacterial cells rupture. So how does the bacteriophage reprogram the bacterial cells to make more viruses? And does the bacteriophage DNA, the protein, or both give instructions to the bacteria? Harshey and Chase inserted the radioisotope of phosphorus 32 into the phage DNA and that of sulfur into the proteins of a separate phage culture. P is not found in proteins but is an integral part of DNA. And S is present in proteins but never in DNA. So when the bacteriophages infected the bacteria, indicating that the phage DNA entered the cells. P32 also be recovered from phage progeny, and this procedure was performed once for the sulfur S35 phages, and the labeled progeny were then allowed to infect unable bacteria. The phage coats remained on the outside of the bacteria while genetic material intact. Disruption of the phage from the bacteria by agitation in a blender followed by centrifugation and allowed for the separation of the phage coats from the bacteria. So, here is the conclusions. The Hershey-Chase experiment helped to confirm that DNA, not protein, is the genetic material, and genetic information is transmitted by DNA only. So these bacteria were lies to release phage progeny, and the progeny of the phage that were labeled with radioactive phosphorus S remains labeled, whereas the progeny of the phages labeled with radioactive sulfur were unlabeled. Thus, the Hershey and Chase experiment helped to confirm that DNA is the genetic material, not protein. I hope you enjoyed this lecture. So keep watching and keep learning. In the comment section, you can tell me how satisfied are you with this biology class, and then please put down a comment under this video because this will help me to improve better. So subscribe my channel like and share this video with your friends, family, and I'll also press a bell icon right next to it. And happy watching. To know more about the biology related topics, then keep visiting educational support. Thank you.